Okay, let me just make sure I'm recording. I was doing a recording last time for an hour and didn't go through. Okay, so it's chapter 11. It's introduction to the nervous system. If you recollect, the job of the nervous system is to maintain homeostasis, which means the constant maintenance of your internal conditions, things like body temperature, blood pH, sugar levels, even though changes may be happening outside as well as inside the body. So here, let's say if you're dehydrated, you need some water. You have neurons in general called receptors, like photoreceptors, send information to the brain. Brain analyzes the information, and then it's going to communicate to a target. In this case, it's skeletal muscles, so that you can uptake the water to drink it. Okay, so they're going to break down the nervous system into certain divisions. One of the divisions here is called the central nervous system. Your central nervous system consists of the brain and the spinal cord. Then the other division is referred to as your peripheral nervous system. And for the peripheral nervous system consists of nerves, which are axons wrapped with connective tissue, sending signals up to the brain for sensory, as well as communicating to targets away from the brain. So you have 12 pair of cranial nerves that communicate to targets in the head, as well as receive sensory from the head. And then you have what's called 31 pair of spinal nerves that serve the rest of the body so that you can receive sensory from the periphery of the body, anything away from the head, and also to communicate to targets in the periphery of the body. Now, targets that you can communicate to are going to be one of three things: muscle, glands, and other neurons. So the muscles that you can communicate to are cardiac, skeletal, and smooth. Just remember the only one that you can consciously, voluntarily control is skeletal. You also communicate to glands. You have glands that make hormones. Those are in general called endocrine glands. And you can also communicate to exocrine glands like sweat glands, salivary glands. And then also part of the peripheral nervous system, should you find cell bodies of neurons located outside the spinal cord, they refer to a collection of cell bodies of neurons called ganglia outside the spinal cord. All right, now again, just to review, the central nervous system consists of the brain and spinal cord, and then the peripheral nervous system that consists of the 12 pair of cranial nerves, 31 pair of spinal nerves. They went ahead and they broke the peripheral nervous system down a little further. So those axons that send information up to the brain are called sensory. Excuse me. So you can receive sensory from targets like inside your muscles, also within the body, touch, temperature, pain, things like that, right? And then you communicate to targets. So that division of the peripheral nervous system where you communicate to targets is referred to as motor. And then for motor, they broke it down to which targets you communicate to. So the somatic part of the motor, you're communicating to skeletal muscle, which is the only target that you can consciously control. The other part of the motor is referred to as autonomic. Think of autopilot. Those targets are consisting of the cardiac muscle, smooth muscle, glands, and other neurons. And then the autonomic got further broken down into these two divisions. One prepares your body for physical activity. It's referred to as sympathetic. And they often refer to it as fight or flight. So in times of threatening situations, you either fight, which is confront, or run away, flight. And in the other part of the autonomic division, where there's no threat, you're conserving energy, you're repairing, you're doing digestion. It's called parasympathetic. And they often refer to it to help us remember as it's doing digestion, relaxation time. Okay. And majority of targets that you communicate to are being communicated by both divisions, the sympathetic and parasympathetic. So let's give an example. Your heart, if sympathetic is communicating to the heart rate goes up, so it could send blood faster to skeletal muscle. And vice versa, if there's no threat, you're doing times of digestion relaxation, then parasympathetic would be in control. That would lower your heart rate. It's just that when one's on, the other is off. Okay. So you're constantly going back between these two divisions at the targets. All right, let's clear all drawings here. Then we're going to look at the support cells, the different types you find in the nervous system. So here you're looking at a support cell. It's called an astrocyte. Astro is referring to star shape. So here you got a star shape support cell. And its job is to help shuttle nutrients from capillaries to the nerve cell and then help shuttle its waste products away from the nerve cell. That's one of its jobs. And the other job you can see is tightly wrapped around the capillary that provides oxygen and nutrients. So it keeps things from easily having access to your brain. It's providing what's referred to as a blood-brain barrier, keeping things that may cause harm to the brain out. Okay, let's erase this. The other thing I want to mention is this astrocyte is located within the brain or spinal cord, which is part of your central nervous system. Now what you're looking at here is a microglial cell, also located in the central nervous system, and it's a white blood cell. So white blood cell's job is to look for anything foreign that may cause harm. That's what it's doing. Now, a third type of support cell here in the drawing, they're called ependymal cells. Now, if you cut into the brain, there's cavities within the brain called ventricles where you have fluid called cerebral spinal fluid, and that fluid provides oxygen and nutrients to nerve cells deep in the brain. And lining the ventricles are these support cells called ependymal cells. So ependymal cells, they have cilia to help propel the fluid so it doesn't just remain stagnant. The other thing the ependymal cells provide is a barrier, so not anything has easy access to your neurons deep in the brain. That is the function of ependymal cells, again, located in the central nervous system. What you're looking at here is another type of support cell called an oligodendrocyte. It's making a lipid substance called myelin and wrapping around the axons of neurons. Axons are the tails of a neuron. So it's this myelin that helps to communicate to targets quicker if the targets are located far away. So neurons that communicate to targets far away, you're going to see myelin. And this cell, the oligodendrocyte, is located again within the central nervous system, which is in the brain or in your spinal cord. Now, we're in the peripheral nervous system, which means outside the spinal cord. There's these type of support cells. One's called a satellite cell. It's similar to the astrocyte in that it helps shuttle nutrients and oxygen to the neuron and also shuttle away waste products produced by the neuron. Another type of support cell is called a Schwann cell. Its job is to make myelin and wrapping around the axons of neurons located within the peripheral nervous system. So they're similar to the oligodendrocytes. It's just they're located, again, Schwann cells in the peripheral nervous system. You might notice in this picture, just take note, not all neurons are going to look the same. We'll get into how they're named in a little bit. So this is our typical picture of a neuron. Notice it did not look the same as the previous picture. So this neuron, this is our typical picture, it's called a multipolar neuron. The way they name this neuron is by how many processes come off the cell body. So here's the cell body. The processes would include these branches called dendrites, where the neuron can be communicated by another neuron by the release of neurotransmitters. The other process here would be the tail of the neuron called the axon. Well, since it has many processes, therefore it's referred to in general as a multipolar neuron. In the previous picture, this one looks like to have only one process coming off the cell body, even though it's actually more than one. Here's the cell body. Here's the process. So this one in general is referred to as unipolar neuron. Uni indicating it looks like there's only one process coming off the cell body. even though it's more than one technically. So another way you can refer to it as is a pseudo unipolar neuron. Pseudo means false. Okay? All right, going back to our picture of the multipolar neuron, going over the features of the neuron, it has again branches called dendrites where other neurons can communicate to this neuron by releasing chemicals called neurotransmitters. And then the neurotransmitters have to bind to chemically gated channels located on the dendrites or on the cell body. Fancy name for the cell body, it's not listed here, is referred to as soma. And then the axon, where the cell body meets the axon, there's like a funnel-shaped region right here. That funnel-shaped region is referred to as the axon hillock. And then portions of the axon that are not wrapped with myelin are referred to as myelin sheath gaps. Or you may see in other textbooks or in your reading nodes of Ranvier, like I learned it. Some person named that, discovered it, got to name it. What's special about the axon hillock is you have different type of channels that will open up in response when you can change the voltage inside the cell. Channels that open up in response to change in voltage are referred to as voltage gated channels. At the end of the axon, where it starts to branch, go in different directions, it's called terminal branches. And at the end of each terminal branch is a bulbous region where it looks like a bulb, and that's where neurotransmitters can be released. That bulbous region is called an axon terminal. Now, what are we looking at here? We're looking at a Schwann cell, which is a support cell located in the peripheral nervous system. The job of the Schwann cell is to make myelin and wrap it around the axon so that this neuron can communicate to targets further away a lot more quicker. So here you see the nucleus of the Schwann cell, and as it wraps the myelin around the axon, forms what's called a myelin sheath. The outer region of the myelin, not labeled here, is referred to as neurolemma. Now, this is showing us channels that you would find on the dendrites or on the cell body, channels that open up in response specifically to neurotransmitters. Because a neurotransmitter is a chemical, the channel is referred to as a chemically gated channel. So here's the neurotransmitter. If it's very specific for a channel, it'll bind to what's called a receptor region, where only that neurotransmitter will fit, like a key to a lock. It has to fit perfect in order to open the channel. So channels that open up again in response to chemicals, you refer to them as chemically gated, or you may refer to them as ligand gated. Ligand means chemical. Now, you may have channels that only open up when you can change the voltage inside the cell to a certain level. Channels that open in response to changing the voltage, or when there is a change in voltage, are referred to as voltage gated channels. So inside the cell, it's typically at rest. The resting memory potential, the voltage is at minus 70 millivolts. If you can change the voltage to about minus 50 millivolts, there are voltage gated channels for sodium that will open. Therefore, they're called voltage gated channels. Once one voltage gated channel opens, if there's one next to it, it will open, causing the one next to it to open. Similar to like dominoes. If one domino falls, they all fall. So in this scenario, if one voltage gated channel opens, the ones next to it will all open. Here's a drawing of a device called a voltmeter, and it is measuring the voltage difference inside the cell and comparing it to outside the cell. Now, inside the cell, you do have ions, both positive and negative. Outside the cell, you also have ions, both positive and negative. However, using this device, it measures the difference. Inside, it's more overall electrically negative when you compare it to the outside. This negativity compared to the outside is referred to as the cell's resting membrane potential. And most cells, when it's at rest, that voltage is at minus 70 millivolts. Now, anytime you have positive ions flow in the cell and you make it less negative, or you could say more positive, the term is called depolarization. So we have a cell and the neuron releases neurotransmitters and opens up channels for positive ions to come in. As positive ions flow in, you're changing the voltage, you're making it less negative. So at rest, it's minus 70 millivolts, but as positive ions come in, it's becoming less negative. That term is called depolarization. Okay, let me erase this. Anytime the cell, for whatever reason, inside falls below minus 70 millivolts, that is going to be referred to as hyperpolarization. How can the cell become hyperpolarized? Is when negative ions come in from outside or when positive ions leave from inside the cell, then it's becoming more negative. That's called hyperpolarization. Now, as positive ions, let's say, come in, if they come in the middle of the cell, they're going to move in directions where it's more negative because opposite charges will attract. We said overall inside it's more electrically negative compared to the outside. So as positive ions flow in, they'll start moving in directions where it's more negative. So if it's in the middle, they'll flow in both directions, the positive ions, as they come in. So let's say positive ions come in, they go in where it's more negative on each side. Now, when you send information to the brain for sensory, whether it's touch, temperature, at some point you may interpret some type of stimuli like touch, temperature as painful. How does the receptor send information to the brain indicating, hey, this is painful, it's starting to cause damage? Well, that's when you create an action potential. What is an action potential? Well, an action potential is the brief reversal of the cell's resting membrane potential from minus 70 to positive 30 millivolts. Okay? So let's say you're sending information to the brain for sensory. Let's say touch. So when you physically push on the receptor to send information to the brain to touch, it makes a few action potentials. Hey, that's not too painful. However, if it starts to be interpreted as painful, like deep touch increasing in pressure, the receptor makes a bunch more action potentials per time. So the more action potentials that occur per time, that tells the brain that's a very strong stimulus, where you probably interpret that stimulus as painful. So as the stimulus increases, whether it's touch, temperature, etc., the receptor makes more action potentials per time, telling the brain it's very strong. Okay, what is the general definition of an action potential? Again, the definition is a brief reversal of the cell's resting membrane potential from minus 70 millivolts to positive 30 millivolts. The nervous system communicates to three targets in general: muscle, glands, and other neurons. If I can create an actual potential inside a muscle from minus 70 to 30 millivolts, the muscle will contract. However, if I create an actual potential of a neuron, the neuron's not going to contract. It will release neurotransmitters. If you create an action potential in a gland, the gland's not going to contract. The gland's going to release whatever it makes. So you can create an action potential in 3 different targets and get 3 different actions. For a muscle, it'll contract. For a gland, it'll release its product. And in a neuron, it'll release neurotransmitters. Okay, so what are we looking at as well here? Let me erase this. Now, when you're causing the target to do an action potential, right? You're well within it. There's a period of time where you cannot get the target to do another action potential. It's similar to if I send you on an errand, I can't get you to do a second errand until you complete the first one. So there's a period of time where the target's doing an action potential and you can't get another action potential at the target. That's called the absolute refractory period. Now, when you come back to rest, let's say at 30 millivolts, what happens to come back to rest? You will have what's called voltage gated channels for potassium to open. They open up at 30 millivolts. And because there's more potassium inside, it wants to go out. You're losing positive ions. You're coming back to rest. The term is called repolarization. So right about in this area here, let me erase. Right about here, you're almost back to minus 70 millivolts, but not quite. But at this point in time, called the relative refractory period, I could get another action potential. I just need a stronger stimulus to create another action potential. What do you mean stimulus? Well, in this case, the stimulus was the release of neurotransmitters onto another neuron. So the first release of neurotransmitters, I'll draw it in red, is shown here. First release of neurotransmitters is the stimulus. So if I want to create an action potential before I come back to rest, right about here, I need more neurotransmitters than I did the first time. So if I can release more neurotransmitters than I did the first time, I could create an action potential at a point called the relative refractory period. It's a period of time where the target's almost back at rest, but not quite. You have enough voltage gated channels for sodium close. You can re-stimulate them by releasing more neurotransmitters. And remember, anytime you fall below rest, that term is called hyperpolarization. So when you're coming back to rest, when potassium's leaving the cell, those channels for potassium are open up a little longer than you need. You fall below rest. Anything below minus 70 millivolts is called hyperpolarization. They will eventually close, and then your pumps, called sodium potassium pumps, and then channels that are just naturally open, called leaky channels, bring you back to your resting memory potential at minus 70 millivolts. All right, this picture is showing us, one, the difference between chemical gated channels and voltage gated channels for sodium. Let's see here. So let's say when you release neurotransmitters onto a target, the first type of channels that open up to the neurotransmitter are called chemically gated channels. Remember that the chemical has to fit perfect on a receptor region to open the channel. If it's for sodium, sodium comes in. Sodium has a positive charge. That means the target's becoming excited. It's becoming depolarized. Now, when you open up chemically gated channels at a dendrite, The positive ions, as they flow in, they have to travel. They have to travel to the axon hillock in order to open up voltage gated channels for sodium. And as they're traveling, some of the positive ions, as they're traveling, are being pumped out by a pump. It's called the sodium potassium pump. This pump pumps out three sodiums, three positive charges of sodium, and simultaneously brings in two potassiums. So if you pump out an extra positive ion, then you're bringing in, you're making the inside negative. It's trying to maintain the inside at minus 70 millivolts. These pumps, similar to, let's say, if you had a boat with a leak, you have a pump, if you want to stay afloat, has to pump the water out. So these sodium potassium pumps are trying to keep the inside at minus 70 millivolts. So as positive ions for sodium travel from the dendrites, these branches, to the axon hillock, they're not all going to make it. Some of them are being pumped out. So this signal, as the positive ions travel, because it gets weak over distance and time as they're being pumped out, we call that a graded potential. Graded means it can eventually be high or low depending on how many ions are pumped out. So graded potentials are when you open up chemical gated channels. The signal over distance and time gets weak because of the pumps. Voltage gated channels are located at the axon hillock. Recollect, if you open one, they all open, similar dominoes falling. So here's a voltage gated channel. So as positive ions come in, it goes to the next channel, it opens, that channel opens, they all open. These channels are very close to each other. So no matter if there's a pump that pumps some of the ions out, the channels all along the membrane still open. So this type of potential is called an action potential. It does not get weak over distance and time. And what type of channels create an action potential? Voltage gated. For which ion? Sodium. Now in the third scenario below, these are where an axon is wrapped with myelin. So you still have a voltage gated channel open, positive ions come in. But because the axon here is wrapped with myelin, you cannot pump out any ions. There's no pump there. And as positive ions come in, it gets more concentrated. It gets very crowded. Think of Fiesta, it's very crowded. You want to get to where there's less people. So as it becomes concentrated, those ions quickly move faster to the next channels, causing them to open up faster. So action potentials don't get weak over distance and time, but they can go faster if the axon is wrapped with myelin. They say myelin insulates the signal. What's the signal? Is the conductance, the influx of sodium ions moving to the next channel, causing it to open faster. Okay, we're looking at a synapse. What is a synapse? A synapse is just a point of communication between a neuron and another neuron. Typically, the one doing the communication first is always referred to as pre-synaptic neuron, and then the one being communicated to is a post-synaptic neuron. So here in this scenario, we got 1, 2, 3 presynaptic neurons communicating to a postsynaptic neuron. Each presynaptic neuron can release neurotransmitters. Depending on where it releases the neurotransmitters will tell you what type of communication synapse that it is. So in this presynaptic neuron, it's releasing neurotransmitters on the cell body. Fancy name for cell body is soma. So this synapse is from the presynaptic neuron's axon onto the postsynaptic neuron's soma. So this synapse is called an axosomatic synapse, from axon to cell body. This other presynaptic neuron up top is releasing neurotransmitters from its axon onto dendrites. So this type of synapse is referred to as an axodendritic synapse, from axon to dendrite. The third scenario down below is called an axo-axonal synapse, basically from axon to axon. The one that has the best chance of creating an ax potential will be the ones right near the voltage-gated channels. Recollect, voltage-gated channels will open up at minus 50 millivolts. If one opens, they will all open all along down the axon. Well, just think, if I open up channels on the dendrites, positive ions have to travel from the dendrites to the axon hillock and still have enough ions to reach minus 50 millivolts. However, they're not all going to make it because of pumps called the sodium potassium pump. So some of them are going to be pumped out. So in order to reach threshold at the axon hillock, I'd have to open up a bunch or a lot of chemically gated channels on the dendrites or on the cell body to keep the pumps occupied and still reach threshold at the axon hillock. So then, the synapse that has the best chance or easier chance creating an action potential is if you can open up chemically gated channels right near the axon hillock. That way you don't give the target enough time to pump any ions out, and you reach threshold a lot faster here if you're right near the axon hillock. All right, this channel opens up in response to a chemical, so you refer to it as chemically gated channel, or like I mentioned earlier, a fancier name would be ligand gated channel. Ligand referring to chemical. Now, because you can immediately have an effect on the target by changing the voltage through releasing neurotransmitters, this effect is referred to as a direct effect. You have an immediate change on the target. That's the advantage, but the disadvantage is that it's not very long lasting. Let's look at a different scenario. Now, in this scenario, you've got the neurotransmitter binding to a receptor protein, but it's not a channel. So here you go through a cascade activating other proteins. You activate an enzyme, activate what's called second messengers, which activate more enzymes to get the overall response at the target. So here, the overall response of the target is to open up channels, but you're not just opening up one, you're opening up many. So the response overall is amplified. In other words, magnified. It's going to last longer. The disadvantage is it takes a while. So when targets respond in this way, where it takes a while, but the effect's amplified by the release of neurotransmitters, this is called an indirect effect. Another effect that can happen at the target besides opening or closing channels is to initiate the production of proteins. And remember, the proteins, the instructions to make them are located in the cell's nucleus called DNA. So you can also have indirect effects to make proteins. We want to recognize what a circuit is. Circuit is just when one neuron communicates to other neurons. There's two types of circuits we want to recognize. One's called diverging, the other one's called converging. This one happens to be a diverging circuit. When you have a presynaptic neuron and it communicates to many, diverge, that's how the circuit is named, diverging. If it were going in the opposite direction where you have many communicating to one, it's coming together, that would be called a converging circuit. Convergence, come together. The discharge zone, you'll notice here that a lot of the axon terminals of the presynaptic neuron are right in the middle here. So you can release a lot more neurotransmitters in this area called the discharge zone. So you're more likely to create action potentials at these neurons in the discharge zone versus the facilitated zone. There's less axon terminals. All right. Before I get to that, let's just look at a converging circuit. So converging circuit, you have neurons sending information from many to one. Give an example, think of an environment. You send sensory that you smell the environment, touch the environment, hear the environment, and the brain comes up and forming an emotion. So you're sending a lot of information to one area. That type of circuit is called converging. This picture here is showing us what a reflex is. Reflexes that you're born with, the response from the nervous system is not from the brain, but located within your spinal cord. So let me just go over real quick why the difference in color here. Anytime you see gray in the brain or spinal cord, that's where you have cell bodies of neurons. Anywhere you see white, those are axons with myelin. Myelin gives the appearance of white. Now, again, a reflex. Let's just say an example. You touch something hot, your immediate response is to pull away from what's causing the pain. That's not determined by the brain, but you still feel the pain. So yes, a stimulus occurs. Let's say it's hot. You touch something hot. The information, one, goes to the brain. You felt it. But the response bypasses the brain. First, you have a receptor sending the information to the brain, sensory. But the response bypasses the brain and next goes to a interneuron. Basically, interneuron means in between. So now the response goes through an interneuron, which is in between the sensory and what's next called the motor neuron. Motor is you're communicating to targets. So after the motor neuron and you're communicating to the target, we generally call effector as a target. This is a somatic reflex. Somatic is referring to that you're communicating to skeletal muscle. You'll see why when we get into chapter 12 or something like that. I'll distinguish how I knew that this was a somatic reflex, because you also have autonomic reflexes that you cannot consciously control. So again, a reflex is a response by your nervous system, not determined by the brain. Step one, a stimulus occurs. Information goes through a sensory neuron, entering the back of the spinal cord. The response bypasses the brain, goes through an interneuron, then out of the front of the spinal cord through a motor neuron, finally to the effector, which is the target. We already saw a converging circuit from many to one. Some circuits may be repetitive, like repeat. So here you're looking at a reverberating circuit where the information is going repetitively through these neurons. Examples, when would you want to repeat something? Well, breathing. Also, things like communicating to muscles that you're involved in walking to repeat the walking pattern. This type of circuit's called, what did they say here? Parallel circuit, which means information is going along collateral channels of neurons that are next to each other in the same direction. So examples of parallel circuits that you'd be using are like in solving problems, mentally thinking, maybe doing some like math calculations. Some definitions here. What is a nucleus? Well, no, it's not the nucleus inside the cell that contains DNA. For whatever reason, in this chapter, nucleus is referring to the cell bodies of neurons located within the brain or spinal cord. And the brain or spinal cord, again, is the central nervous system. Vice versa, let's say you have a collection of cell bodies of neurons, but outside the spinal cord, then it's referred to as ganglion. That will be located in the peripheral nervous system outside the spinal cord. When you have a collection of axons wrapped with connective tissue and you're in the brain or spinal cord, it's referred to as a tract. If you have a collection of axons wrapped in bundles, sending information to targets or receiving sensory that you need to send to the brain, but it's coming off the spinal cord. It's called nerves. Those include the 12 pair of cranial nerves that serve the head, and then you have 31 pair of also spinal nerves that serve the rest of the body. I think I did write this out earlier in the beginning of this chapter. Okay, let's move on. All right, let's have a look at these neurons. Let me make sure, let's stick to that picture. So how do we name neurons in general? Well, if it looks like there's many processes coming off the cell body, like the dendrites in the tail, there's many. It's referred to as a multipolar neuron. If it looks like there's only two processes coming off the cell body, then it's referred to as a bipolar neuron. If you happen to see typically one process coming off the cell body, you refer to it as unipolar, even though typically it's more than one theoretically. So you could also refer to it as falsely one, which is pseudo-unipolar. Now, as well, even though they may not look the same, they're all going to have similar features in that they're going to have an area of dendrites where another neuron can communicate to it through the release of neurotransmitters. Two, they're all going to have a cell body. And three, each neuron type will have its own axon terminal to release its neurotransmitters to communicate to targets. And last but not least, you may have multipolar neurons, bipolar neurons that are falling in those categories, but yet they don't look the same. So here you got two types of multipolar neurons. You can see that they have many processes coming off the cell body. Just depends on the location they're found in that gives them their appearance. So these multipolar neurons are located in the cerebellum part of the brain. This one here called pyramidal cells located within your brainstem. Bipolar neurons, you can see two processes coming off the cell body. This type here called an olfactory cell, you'll find in areas where you're going to do smell, up high off your ethmoid bone. Olfaction means smell. And then the retinal cell, you'll find within the eye, the retina helps convey information dealing with vision. The unipolar neuron, you'll find it outside the spinal cord where you got a cluster of cell bodies outside. Remember, that's called ganglion. So these type of neurons, the cell bodies are located in what's called dorsal root ganglion. So just to kind of give an area where each type you may find and what location. Multipolar neurons, they communicate to targets. So leaving the spinal cord here, they form the motor neurons that communicate to your skeletal muscle. Bipolar neurons, one of the areas you can find it is within the retina of the eye. They help pass along information, sensory, dealing with vision to the brain. And then the unipolar neuron, these unipolar neurons are the ones that send sensory, and they enter through the back of the spinal cord to send sensory up to the brain. Sensory like touch, temperature, pain, any type of sensory you can think of. All right, what do we have here? So some neurons are trying to excite versus some neurons may be trying to inhibit a target. A neuron that tries to excite is referred to as excitatory postsynaptic potential. Neurons where they try to inhibit are inhibitory postsynaptic potentials. All right, so how would a neuron try to excite a target? Well, by making the insight less negative, right? Called depolarization. So the way you can depolarize a target is to release neurotransmitters that will open up chemically gated channels for sodium to come in, which is positive ions. Yes, a little potassium leaves, but if you have more positive ions come in, the overall target is being depolarized. So anything above minus 70 millivolts is called depolarization. All right, how could you inhibit a target? By making it more negative, go below minus 70 millivolts. That would be a scenario where you can release neurotransmitters specifically for channels that allow chloride to enter. Chloride has a negative charge, and because there's more of it outside, it wants to come in. So as negative ions come in, that means you can hyperpolarize the target, make it more negative. The other scenario where you can make it more negative is by opening up channels for potassium to leave. So here you got a chemically gated channel for potassium. When it opens, there's more potassium in and it wants to go out. And the charge on potassium is positive. So as you lose positive ions, you're making it more negative. You can also hyperpolarize a target that way. Voltage-gated channels, recollect, these are the ones responsible for creating our action potential from minus 70 millivolts, not positive, to 30 millivolts. If one opens, they all open. So when does the voltage-gated channel for sodium open? When you reach a certain level of voltage change, about minus 50 millivolts. One opens, they all open. After 30 millivolts, the target's done what it's going to do. If this were a neuron, it'd be releasing neurotransmitters. And now wants to come back to rest. So at 30 millivolts, the next type of channels that will open up are voltage gated, but for potassium. Potassium has a positive charge, so when these voltage gated channels open up at 30 millivolts, they allow potassium to leave. As positive ions potassium leave, you're coming back to rest. The term for coming back to rest is called repolarization. All right, this is a table going over the different neurotransmitters produced by neurons, okay? I'm not going to go over each individual one. I'm just going to bring to your attention that it's not the chemical that determines what happens at the target. It's the receptor type that the chemical binds to. So remember, it's not the chemical neurotransmitter that tells me what happens at the target. It's the receptor that's binding the chemical that tells me what happens. Think of a key to a lock where the key is the chemical, the neurotransmitter. Maybe one key opens the door where you go to another door, it doesn't open it, it closes it. So to give you an example, acetylcholine was the neurotransmitter that when you release onto skeletal muscle, it'll excite it. But if you release acetylcholine on your cardiac muscle, it doesn't excite it, it inhibits it. So that tells me there's two different types of receptors binding the chemical. One's called nicotinic. These are the ones you find on skeletal muscle that cause excitation. And the cardiac muscle, the receptor is called muscarinic. Those are the ones you find on the heart, which would inhibit the heart. The other thing I want to bring to your attention is some drugs. How do they work? Some drugs just mimic the neurotransmitter. It's like having a copy of a key. Does the same thing when it binds to the receptor. Vice versa, some drugs can block a neurotransmitter by covering up the receptor region. Other drugs may cause the target to be more sensitive to the neurotransmitter or more responsive. How? By causing the target to create more receptors. Some drugs may cause the target or the neurotransmitter on the target to last longer. Typically, neurotransmitters are broken down by enzymes, and therefore the channel will close. But if you don't have the enzyme around, the neurotransmitter will last longer. So some drugs may inhibit enzyme production, therefore leaving a neurotransmitter on the receptor a lot longer, therefore having an effect longer at the target. Anyway, I don't have time to go over each individual one. Even here, ATP can act as a neurotransmitter to invoke pain. Typically, when cells die, they release their contents, maybe like ATP, where it binds to neurons, indicating something painful is occurring, causing death to tissue. Even gases can work as neurotransmitters like nitric oxide you find in drugs like Viagra, where they can cause blood vessels to dilate so that men can have an erection, causing the vessels in there to dilate. Okay, so if you want in your own time, look over the table here at the different types of neurotransmitter production. We already went over multipolar neuron, bipolar neuron, and unipolar neuron. Now, what am I looking at here? You're looking at a pump that helps try to maintain the inside at minus 70 millivolts. It's called the sodium potassium pump. It tries to pump out, well, it does pump out 3 sodiums and brings in 2 potassium. If you can pump out 3 positive ions and only bring in 2, you're making the inside more negative. Okay, so that's helping to maintain what's called the resting memory potential at minus 70 millivolts. Similar to a pump, let's say in a boat, if the boat had a leak, you'd want to pump out the water to keep the boat afloat. You do also have channels that are just naturally always open called leaky channels. So as I go down here, let me get the marker out. You got some channels just naturally open, leaky channels, one for potassium, the other for sodium. Because there's more sodium outside, it wants to come in. And because there's more potassium inside the cell, it wants to go out. All these 3 mechanisms working together, which are the leaky channels for potassium, leaky channels for sodium, and your sodium potassium pump. All 3 work together to maintain the inside at minus 70 millivolts, which is referred to as the cell's resting membrane potential. What you're looking at here is a presynaptic neuron, first one, communicating to a postsynaptic neuron. Okay, so step 1, the presynaptic neuron voltage-gated channels of sodium are opening, creating an action potential. Remember, if one opens, they all open. Positive ions are fluxing in. Eventually, you cause the opening up of a voltage-gated channel for calcium. Okay, let me magnify here and get the pin out. Calcium, we need calcium. As calcium comes in, it guides these vesicles. These vesicles are called synaptic vesicles. These are the vesicles that contain the neurotransmitter. So calcium guides the vesicle to fuse its membrane into the membrane of the axon terminal. Once the vesicle's membrane fuses in with the axon terminal, that causes the neurotransmitter to be released. The neurotransmitter will cross this space. There's always a little space between the neuron and the target. That space is called the synaptic cleft. And like a key to a lock, if it fits perfect, it knows it's in the correct area. The neurotransmitter will bind to a receptor region on this chemically gated channel. It happens to be specific for sodium. Let me erase all that. And as sodium positive ions come in, the inside's becoming depolarized. If I reach a certain level of change, about minus 50 millivolts, the next type of channels that will open up are voltage gated for sodium. If one opens, they all open, okay? Creating our action potential. At some point in time, the neurotransmitter has to come off. What helps take it off are enzymes. Enzymes within the space called the synaptic cleft will break the neurotransmitter apart. Or just over time, the neurotransmitter can diffuse away from the receptor region. Not all neurons are trying to excite. Some will be trying to inhibit. Okay, so here you have some presynaptic neurons that are trying to excite. They're telling the target to fire, fire. In other words, they're trying to create an action potential at this target. Other neurons may be trying to inhibit by hyperpolarizing the target. Typically, the ones that win out are the majority. It's like a vote. the majority have their say. So those neurons, there's more of them trying to excite. That's probably what's going to happen at the target. It's going to be creating an action potential. All right, so what do we got in this drawing here? So here you got a presynaptic neuron. Looks like it's trying to excite a post, releases neurotransmitters that open up chemically gated channels for more sodium to come in. As more positive ions come in, the target's becoming depolarized. So this type of communication is referred to as an excitatory post-synaptic potential. The other scenario, this neuron, the presynaptic neuron, is trying to inhibit the postsynaptic neuron by releasing neurotransmitters to make it more negative inside. It can do that by releasing neurotransmitters to allow negative ions to come in, like chloride, or to open up channels to allow potassium to leave. Either scenario will cause the inside to hyperpolarize. So this type of synapse where the target's being inhibited is referred to as an inhibitory postsynaptic potential. Hold on, let me just make sure got everything. I believe so. So this is the end of the chapter, and I'll convene back when I go to the next chapter. Let's see. I'm gonna end this meeting for everyone.